Did you know that every 40 seconds, someone has a heart attack, which is 805,000 heart attacks annually? Out of these 805,000, 647,000 people die every year of heart attacks, making it the leading cause of death. And familial hypercholesterolemia, a heart condition, makes it 20 times more likely, which is why familial hypercholesterolemia is one of the most impactful conditions in the entire world, and is why I use genomics to study it. That leads to my project, From Trend to Target, discovering mutation patterns that shape familial hypercholesterolemia pathogenesis. My name is Nikhil Puttamraju, and I'm here to present this topic to you. So, a quick about me. I'm a high school junior at Paradise Valley High School Crest Bioscience in Phoenix, Arizona. So it's really hot out here, and heart conditions are common. I'm really interested in cardiovascular medicine, public health, and cell biology. And I have a personal experience with FH, being diagnosed when I was 15-year-old, which makes this very interesting for me to analyze through my field of interest, cardiovascular genomics. My mentor was Ron Melde, assisted throughout this entire project, and I thank him a lot for it. So let's get into it. To begin, FH is an inherited cardiovascular disorder that leads to abnormally high LDL cholesterol levels, which are often called bad cholesterol. This buildup stems from pathogenic variants in genes such as LDLR, APOB, and PCKS9. What makes FH particularly dangerous is that it often causes premature atherosclerosis and raises the risk of early heart attacks, even in people who otherwise appear healthy. Both coding and non-coding variants of genes can drive this disease by disrupting cholesterol uptake, receptor recycling, or metabolic regulation. Understanding these mutations is critical because it not only informs treatment strategies for FH, but it also sheds light on cholesterol regulation as a whole, which impacts millions of people worldwide. This brings me to the central question of my project. What trends and patterns exist in variants associated with FH, and how can they be used to develop new treatments? To answer this, I broke the project into several goals. First, to categorize mutations driving FH, distinguishing between coding and ongoing variants. Second, to explore which biological pathways are most enriched for disruption. Third, analyze mutation spectrum patterns such as transitions, transversions, and indels, which I will get into in the future. And finally, to connect all of these findings back to their real-world implications, from diagnostics to treatment development. To investigate this, I gathered FH-associated variants from large-scale genomic databases like ClinVar, Ensemble, and DBSNP, supplemented with variants reported by 4 peer-reviewed FH studies, giving me a data set that I collected personally of over 12,000 total variants. Using tools such as Ensemble's Variant Effect Predictor, GO databases, and RStudio, I carried out multiple analyses: mutation spectrum characterization, variant effect prediction, and gene ontology enrichment. This combination of data and computational approaches allowed me to not only quantify mutation types, but also understand their biological significance. Let's start with variant consequences. The majority of FH-associated variants were classified as missense, meaning they alter a single amino acid in a protein. This is especially important in FH, since even small changes to LDLR, APOB, or PCKS9 can severely impact LDL binding or clearance. Another striking finding was the high proportion of nonsense mutations and frameshift variants, which tend to have much more severe effects. These mutations essentially cut short or scramble the proteins, frequently rendering them non-functional. Their enrichment in FH suggests that a total complete loss of protein function is a key driver of the disease. Importantly, this also means that therapeutic strategies must consider not just modifying protein activity, but sometimes compensating for a total lack of the protein's function. Looking at a biological process gene ontology enrichment, the most enriched items were fatty acid metabolism and cholesterol metabolism. This confirms the direct role of FH mutations in disrupting pathways at the heart of lipid regulation. Increasingly, I also found enrichment in processes related to lipid processing and trafficking, indicating that it's not only cholesterol uptake that is impaired, but also how cells handle and distribute lipids more broadly. These disruptions could explain why FH patients often have widespread metabolic complications beyond elevated LDL. Next, I did a gene ontology enrichment on molecular function. In this function, the strongest enrichment was lipid binding and cholesterol transporter activity. This suggests that many FH-associated mutations directly interfere with protein-ligand interactions, particularly those involving LDL and its receptor. Essentially, the molecular machinery designed to clear cholesterol is destabilized at its most fundamental interaction points. The implication here is that small molecule therapies or biologics that restore or stabilize these binding interactions could be a highly effective approach to treatment. Next, the third gene ontology enrichment, cellular components. On the cellular component side, enrichment appeared in plasma membranes and endocytic vesicles. This makes sense because LDL receptor activity depends on proper membrane localization and endocytosis. Mutations that disrupt these processes likely prevent receptors from reaching the cell surface or recycling efficiently after LDL uptake. This emphasizes that FH is not only about the presence of receptors, but also about their dynamic trafficking. Understanding this could help refine therapies that improve receptor recycling or stability, rather than just receptor expression. The final and third thing that we studied was mutation spectrum analysis, where we understood the types of variants. Turning to this mutation spectrum analysis, one of the first things I observed was that the majority of variants were single nucleotide variants rather than large insertions or deletions. Many of these occurred in non-coding regions, which suggests that regulatory mutations may play a bigger role in FH than previously recognized. This is important because non-coding mutations don't alter the protein itself, but instead affect how much, when, or where the protein is expressed. That means diagnostic pipelines that only look at coding sequences may miss a significant fraction of disease-causing mutations. Next, when comparing transitions to transversions, I found a clear bias towards transitions, with roughly twice as many transitions as transversions. This is consistent with general human mutation patterns, but has an important implication. FH mutations largely follow baseline mutational processes rather than being driven by unique or disease-specific mechanisms. In other words, the high burden of pathogenic variants is not because FH genes are unusually prone to rare mutation types, but because even common mutational events in these genes have major consequences for LDL regulation. Mutation spectrum for AT and GC cells. Additionally, interesting results came from analyzing these trends. Mutations were about 60% more frequent in GC guanine cytosine sites, consistent with the known instability of CpG dinucleotides. This highlights a sequence context bias in FH, where certain regions of the genome are inherently more vulnerable to mutation. Clinically, this matters because it means that specific hotspot regions within FH genes are likely to accumulate mutations across patients, which could guide targeted screening efforts. When analyzing indels, also known as insertion or deletions, I found they were much less common than single nucleoside substitutions, but showed strong positional bias. Specifically, indels were clustered in repetitive or low complexity regions, which suspects that many may arise from replication slippage. Many of these regions overlap with microsatellites, further linking FH mutational mechanisms to genome instability. While rare, these indels are especially disruptive, often causing frame shifts that destroy protein functions. This underscores the need to carefully evaluate indel variants in clinical sequencing, even if they occur infrequently. Now, stepping back, what do these findings mean for the real world? First, diagnostics. Current FH testing often focuses on only coding regions, but my findings show that non-coding and regulatory mutations also play a critical role. Expanding diagnostics to capture these variants could improve detection rates, especially in patients with strong clinical FH, but non-coding regions. Second, precision medicine. Integrating genetics findings with pharmacogenic or environmental factors could help redefine risk protection and treatment tailoring. Third, therapeutics. The data suggests that opportunities to guide next-generation interventions, from CRISPR-based editing to small molecule therapies that stabilize receptor ligand interactions. Finally, population screening. Understanding sequence biases and hotspots can make genetic testing more efficient, especially in families with inherited FH. Looking forward, there are several next steps. One is a functional validation, experimentally confirming how non-coding variants alter gene expression and splicing. Another is transcriptomics and splicing models, using RNA sequences and computational approaches to map how these mutations affect transcripts across the genome. Personalized therapy is also a major opportunity. Designing interventions tailored not just to the gene affected, but to the specific type of variant, whether regulatory, coding, or structural. Ultimately, these efforts can pave the way for more precise, patient-specific treatments, bringing us closer not just to managing, but potentially curing FH. All of my research is built on a lot of published work, as well as the data analysis I performed. I owe a huge thanks to the researchers who laid the groundwork that I could build on. And if there's one message I hope you take away, it's this. Sometimes the answers we're looking for aren't in the obvious places. They're hidden, waiting for us to dig deeper. And when we do, the impact can be life-saving. Thank you for listening. Any questions?
Fantastic presentation. Thank you so much. As a reminder, if you have some questions, feel free to add them in the Q&A tab. It might be under the More section in your Zoom. But to get us started, I was kind of curious, do you think some of the insights from the FH mutations that you found might also apply to some other cardiovascular diseases or other diseases in general?
Definitely. As we can see back in I'm just going to go back in my slides really quickly. In the sections about gene ontology and enrichments, these cellular components that are found in familial hypercholesterolemia, such as lipid binding and cholesterol transport activities, are found in so many different heart conditions or vascular conditions, such as lipoprotein A elevation or homogeneous hypercholesterolemia, or just any cholesterol-based vascular condition. So I think that these research over here can be applied or specified for any vascular condition, whether strokes and cerebrovascular or heart conditions.
Great. And then I guess I'm also kind of curious too. Maybe how do you think tools like CRISPR or AI might help in kind of understanding the effects that these different variants might have?
Well, I feel like analyzing the effects can already be done with a lot of statistical software. However, AI and CRISPR can help with future applications. As I said on my slide over here, one of my most favorite and interested pieces of future direction is personalized therapy, where we can have specific medicines tailored for specific people's genes. And the way to analyze this would be through CRISPR modeling and AI tools, where we could analyze their entire genome with our new genetic technologies using CRISPR, and we can personalize our medication to target specific genes that cause their FH or any other conditions. So it's more of an application point of view rather than a usage in current methods.
Fantastic.